So class, let's talk very briefly about the differences between hemoglobin and myoglobin. We can also talk about some of the similarities. These are two proteins that deliver oxygen to the tissues. Hemoglobin is primarily localized in the red blood cell, also known as the erythrocyte. And myoglobin is primarily the oxygen delivering protein in muscle. Okay, so myo is Greek for muscle. So these are O2 carrying proteins. They carry oxygen. They are proteins, so they will have a primary, secondary, tertiary structure. And in the case of hemoglobin, we have a classic example of quaternary structure embedded on top of the primary, secondary, and tertiary structure. And both of them have similar functions, but the way they bind oxygen is going to be very different between hemoglobin and myoglobin. All right, so the intricate details are actually left for you to decipher in the PowerPoint notes and the notes that I handed out during class. But let's just talk very briefly about oxygen binding and the oxygen binding kinetics. So myoglobin actually would just be a simple monomer that looks like this. Okay, I'm going to depict one single independent protein folding unit as a monomer. It's actually very alpha helical rich. I think it's about 80% alpha helix or alpha helical in terms of its secondary structure. And all the rules of protein folding still are in place. That is, in the interior of this compact protein, folded protein, you'll probably have hydrophobic amino acids. And on the exterior, facing the cytoplasm, you'll have hydrophilic amino acids or amino acids with polar side chains. So myoglobin is a single independent unit, and what actually binds the oxygen is a prosthetic group known as heme. So if you consult the PowerPoint notes in class, you'll see the very intricate structure of heme. It consists of a porphyrin ring. So this really embeds for us the example of many proteins, in order to convey their specific function, need some other ancillary components, need some other ancillary molecules. In this case, in order for myoglobin and hemoglobin, for that matter, to bind to oxygen and deliver oxygen, it does so through these heme groups. And the heme has an iron +2 coordinated with the porphyrin ring. Okay, so this also is the same prosthetic group, heme. The oxygen is coordinated by the iron, so that's an inorganic chemistry phenomenon. Some amino acids play a very critical role in making sure the oxygen stays bound to or coordinated to the iron. The proximal histidine, the distal histidine, very conserved amino acid sequences. Besides many others, not just histidine, but histidine sort of anchors that in place. So we have to come to one realization in our study of biochemistry, is that if it's important, it tends to get conserved. Okay, whether it's myoglobin from whale, and I think the first crystal structure of myoglobin came from whale, or whether it's from myoglobin from muscle, rat muscle, or even human muscle. If it's important, it tends to get conserved all throughout the domains of life. So that histidine, the proximal and distal histidine, as well as some other amino acids that are important in coordinating that oxygen to the iron, they get conserved throughout all domains of life. All right. So continuing on, myoglobin is simply a monomer, so it's not really exciting. Hemoglobin, the excitement really comes in because it's not just a single independent unit, but it's actually a tetramer. Okay, so that's one of the main differences between the two oxygen-delivering proteins in higher advanced eukaryotes. Okay, so actually this is called the alpha-2 subunit. This is diagonally to it is the alpha-1 subunit. This is the beta-2 subunit. This is the beta-1 subunit. All right, so just as before, each of these subunits have about 80% or so alpha helical secondary structure. In fact, these two proteins are alpha helical rich proteins. And what stabilizes these interactions, subunit to subunit interactions, is quaternary structure. The nonpolar, polar interactions, all those things stabilize these subunits. So instead of having one heme, you're going to have four hemes. So I'll just have the hemes kind of drawn like this. So you have theoretically 4 times the bang for your buck, okay, in hemoglobin. So cellular localization, the fact that this is a tetramer versus a monomer, both of them have the same prosthetic group, but this is a tetramer, whereas myoglobin is a monomer. So what are the consequences of having a tetramer? Again, more specificity and more theory that you should know is going to be in your textbook and in the notes that I handed out. But let's just have a very peripheral discussion for now of the consequences of hemoglobin being a tetramer as opposed to myoglobin being a single monomer. All right. One of the things that is a stark difference is the ability to bind oxygen. That is, the oxygen binding kinetics between hemoglobin and myoglobin are very different because one is a tetramer and one is a monomer. So for the oxygen binding kinetics, for myoglobin, if we have on our x-axis the pressure of O2, usually that's given in units of torr, but it can be in atmospheres or any other unit of pressure. And on the y-axis, I'm just going to label this as percent saturation O2. All right. So how much oxygen do you need to get myoglobin, which is a single independent folding protein with a tertiary structure that comes along from its secondary and primary structure? How much does it take? How much oxygen pressure does it take for it to reach 100%? Well, if you increase the oxygen pressure to something, an absurdly high amount, the oxygen will always be ubiquitous, will always be present, and it's just going to saturate the heme, and you'll have complete full oxygen binding. So, you know, the more oxygen concentration you have, in this case oxygen is a gas, so instead of using concentration, we'll use the term pressure. But if you have a lot of oxygen pressure around, there'll be more oxygen available to bind heme, to chelate that iron in the porphyrin ring. So the way this binding curve actually works is that it looks like this. Okay? So that is what the binding curve of oxygen to myoglobin actually looks like. Okay? So what is the name of this curve? All right, this curve actually has a name, and it's called hyperbolic. Okay, if I can spell it. Okay, a hyperbolic curve. Right. So what are the implications of a hyperbolic curve? Well, one implication is if you raise your oxygen concentration or oxygen pressure so sufficiently high enough, eventually all of the heme sites on your myoglobin molecule will be bound with oxygen. All right. So more oxygen around, more likely it's going to go in and bind to myoglobin. All right. What we're really interested in is 50% saturation. the pressure of O2 that's required to saturate 50% of the sites in myoglobin. All right? So that varies whether you have arterial blood or venous blood. So I have the statistics for you. It's not something you have to memorize or be, keen about, but I just put them here for the more interested student. For the arterial blood, this P50, okay, P50 is when 50% of the binding sites of heme in myoglobin are saturated with oxygen. That P50 is going to be 100 torr. Okay, so the pressure of O2 for 50% saturation is going to be 100 torr for arterial blood. And for venous blood, the pressure of O2 to get to 50% saturation is around 30 torr. Okay? So, I'm not going to go into terrible details about this, but venous blood, has a much lower 50% saturation than arterial blood. This means that in venous blood, myoglobin is almost always saturated with oxygen. Almost always. Probably not the case for arterial blood. You may have some myoglobin molecules in arterial blood that may not have oxygen because it takes 100 pressure of oxygen, 100 torr pressure of oxygen to get 50% of those sites saturated. All right, so myoglobin, let's just summarize it right here. One polypeptide, one binding site, one polypeptide chain, one binding site, one prosthetic group. Okay. By the way, venous blood is blood that passes through the peripheral vessels. Arterial blood is blood that has passed through the lungs. Okay, so this is blood, or the arterial blood that's ready to boost oxygen levels to sustain the peripheral organs. So that's myoglobin. Now let's go to its sort of complicated cousin, hemoglobin. Right, again, this is going to be a rush explanation. Okay, more specifics are going to be in your textbook and in the notes that I made out that was handed out in class. So hemoglobin is a tetramer, alpha 2, beta 1, beta 2, alpha 1, quaternary structure, and one of the things resulted from hemoglobin being a tetramer is its oxygen binding kinetics are different. So we'll have the same graph. With the percent saturation O2 on the y-axis and the pressure of O2 on the x-axis. The bindings kinetics, or how fast oxygen binds for hemoglobin, actually takes the form of this shape. Okay? It is not hyperbolic, but it is sigmoidal. It actually has a shape that looks like the letter S. Sigmoidal. All right? A sigmoidal shape. Now, take a look at a sigmoidal shape here. We have the same situation with myoglobin. If I can just zoom out here. We have the same situation with myoglobin, and that situation is that if you increase the oxygen concentration, or oxygen pressure, I'm using those two terms interchangeably here now, if it's high enough, all four sites of the heme prosthetic group in hemoglobin will be saturated. Okay, that makes sense. A lot of oxygen around, and that oxygen will go and saturate your protein. It will saturate the system. Once again, we're interested in 50% as our cutoff. 50% saturation occurs around here. Okay? And it will differ whether it's arterial blood or venous blood. Okay? So, I want you to look at this region of the curve here, okay? If I were to take this blue pen and look at this curve from here to here, that region of the curve tells us something very special about the binding of oxygen, okay, at this pressure of O2. At this pressure of O2, the saturation is here. But if we increase the pressure of O2, the saturation really skyrockets. Okay, it goes from here to here. We increase it a little bit, we go up to here. Ultimately, we increase it to such a level, and then it's going to have this skyrocketing effect. Okay, you see this very steep slope. Okay, it's very steep. Okay, it's very high. Imagine climbing, if you're a human climbing. Okay, you're pretty much going upward, and you're going upward in a very high incline. Okay, what I'm trying to tell you is that when oxygen binds, okay, it's not a linear relationship. Okay, the relationship actually becomes multiplicative. All right, oxygen, increase it a little bit, the percent saturation goes up. Increase it a little bit more, it goes up this much. If you increase it a little bit more, it goes up this much. If you increase it a little bit more, it goes up this much. Okay, so this region of the curve, okay, illustrates a phenomenon known as cooperativity. Okay, cooperativity. That is, the first oxygen molecule binds to one hemoglobin molecule. The second one binds with greater affinity. It's become easy. It becomes even easier for the third oxygen molecule to bind. And by that time, the fourth oxygen molecule, it's very easy. The affinity is very high. That is what this region of the sigmoidal curve is actually telling you. It's telling you that the oxygen binding affinity increases as the first oxygen binds, the second oxygen binds easier, the third oxygen binds even easier, the fourth oxygen binds the easiest. Okay, so what it's actually saying is that when there's an oxygen molecule that binds to the first subunit, there must be some sort of conformational change that gets translated to beta 1 that makes it more amenable to pick up oxygen, which in turn causes a transformational change to occur in beta 2 to make it more amenable to binding oxygen. And then by that time, there's more conformational changes that occur so that the fourth oxygen binds very easy. So the conformational change gets transferred from subunit to subunit so that the fourth oxygen molecule binds very easy. Okay? Naturally, if you increase it to such a high level, you reach saturation kinetics. Okay, this point here is called an asymptote. Asymptote means don't touch me. Okay, so you increase the oxygen even more, nothing's going to happen. Same thing here with myoglobin. Okay, it has an asymptote. So this cooperativity is very important. I think in lecture I talk a little bit about that. It's definitely in our notes. One thing that is also a little bit more complicated about hemoglobin is that each of these subunits, alpha 2, beta 1, beta 2, alpha 1, beta 2, alpha 1, each of these subunits exists in two states. Okay, we have an equilibrium between the R state, which is relaxed, and that's in equilibrium with the T state, which is abbreviated form of tense. The T state is a conformation. Let me redraw my hemoglobin. Okay, here's my heme group. All right, very, very crude, obviously. Okay. Each of these subunits, alpha 2, beta 1, beta 2, alpha 1, can exist in the R state, which stands for relaxed. And it's a three-dimensional conformation of the molecule that favors the formation of oxyhemoglobin, or the form of hemoglobin that has high affinity for oxygen. The T state, like I said before, stands for tight, and that is a three-dimensional conformation of the subunit of hemoglobin that favors the deoxyhemoglobin form. Hemoglobin form. Okay, tight is the T state, so that's a very specific three-dimensional arrangement that the T state is in that favors oxygen dumping. R is a very specific three-dimensional structure of the subunit that favors the affinity of oxygen, or it favors oxygen to go and bind to heme of hemoglobin. Okay, so there's different models of this. You can have all 4 subunits T, you can have all 4 subunits R, you can have RT, RT, RR, RT, T, RT. You can have different variations of this. Okay, that is one of the facets of cooperativity. All right. In fact, there's models of what we call allostery. If you look at your homework, I gave you an assignment where you have to look at your book and mention about the different models of allostery. There's the Mono model. There's the Koshland model of allostery that basically tells you how the cooperativity can get translated across subunits. So allostery is very important. Cooperativity is very important. Allostery actually stems from this curve, okay? Because when you have a sigmoidal curve, not just for hemoglobin, but for other proteins in the cell, this curve of binding of something, of a ligand to a protein, this curve that is sigmoidal not only implies cooperativity if it's a multi-subunit protein, like the tetramer of hemoglobin, but it also implies allostery, which means that you could regulate how well oxygen binds to the heme in hemoglobin. I liken it to the contrast on your television set. You can have the contrast all the way up or all the way down, halfway in between, in the middle. So it's more of a fine tune, okay? And allostery implies that some molecules can come in here and regulate the ability and regulate the affinity of oxygen binding to the heme prosthetic group of a hemoglobin subunit. Okay, so what are these, what I will call allosteric effectors? Okay, well, there are several allosteric effectors that can compete with oxygen binding to heme, such as carbon monoxide. Okay, that's the basis for carbon monoxide poisoning. Carbon monoxide has 200 times the affinity of towards heme than oxygen. So obviously, if there's carbon monoxide exposure, it's going to bind to the heme group in hemoglobin much more tighter and with much greater affinity than oxygen. So actually what that does is any exposure to carbon monoxide, let's say it gets to one of these subunits, okay, it's actually going to favor the shift of the equilibrium more towards the deoxyhemoglobin form. You'll have more of these subunits in the T state, and you're not going to have oxyhemoglobin around. And the net result is, and furthermore, carbon monoxide, it's 200 times more affinity. It's hard to get off. That dissociation between oxygen and deoxyhemoglobin gets really messed up. So you can even consider carbon monoxide as an allosteric effector, something that competes with oxygen and changes this R to T equilibrium. But what about things that are more pertinent to the cell? Because how does the cell regulate whether to have full oxygen binding, that would be in the R state, or to be an oxygen dump and just dump oxygen. Obviously would want to dump oxygen to vigorously exercising muscles, because myoglobin cannot do the job entirely by itself. So what are these some of these allosteric effectors? Okay, so I have a little mnemonic that I use to determine allosteric effectors. All right, so you're going to have to learn the specifics of the mechanism and more of the nitty-gritty in your lecture notes. But this is just some allosteric effectors that change or influence the binding characteristics of oxygen to hemoglobin. So remember the sigmoidal binding curve for oxygen binding to hemoglobin. Remember, we imply that this means cooperativity, and also it means allostery. We have a couple of things here, 3 molecules that shift this curve to the right. Okay? And when it shifts it to the right, I'll just keep this as green. When it shifts this curve to the right, okay, what happens to the affinity of oxygen to hemoglobin? Okay, so what do? We look at the 50% saturation mark. Okay, 50% oxygen saturation. Okay, this is without any allosteric effectors in purple. Here is that, and then shown in green is 50% oxygen saturation as a result of some allosteric effectors. Okay, so what does these 3 allosteric effectors do that I'm going to illustrate to you now? So 50% without anything, okay, the pressure of oxygen is here. Once you add these allosteric effectors that I'm going to now, this is a mnemonic. Don't write it on the exam. This is just to help you memorize. Write on cadet, okay? These 3 things, E stands for exercise, so that's not really a molecule. T stands for temperature, that's not really a molecule. These 3 things actually shift the oxygen binding curve, the sigmoidal curve, to the right. And in effect, they increase the oxygen required to achieve 50% saturation. So what are they doing to the R to T transition? Okay, they're facilitating an oxygen dump. Okay, so we are actually shifting things more towards the deoxyhemoglobin state. All right. Why do I say this shifts? These shifts the subunits more towards the T conformation, towards the deoxyhemoglobin, as opposed to the oxyhemoglobin state. Again, look at the curve. Okay, in the presence of the allosteric effectors, it takes more oxygen to achieve 50% saturation. Okay, so the affinity of hemoglobin to oxygen has actually been reduced. Okay, it's been reduced because it takes more oxygen concentration to achieve 50%. So you're going to really need this much oxygen to overcome the fact that you have these 3 things to achieve that same 50% oxygen saturation to heme of hemoglobin, as opposed to without these 3 allosteric effectors. So what are these things? Okay, so cooperativity, allosteric effectors, write on cadet, commonly used in medical school. CO2 is what C is. A stands for acid. and D stands for diphosphoglycerate. Okay, so this molecule will bind to hemoglobin and shift this curve to the right, favor the T state, causing more oxygen needed to get to that 50% saturation. Acid, which is nothing more than H+, so if there's acidity around, it's going to, or protonation of hemoglobin will favor more oxygen to achieve 50% saturation. Diphosphoglycerate is a very interesting molecule. This actually comes from glycolysis. Okay, so glycolysis is in the last portion of the course. And this is very fascinating to me, but in the glycolytic pathway, we take glucose, and in about 10 steps, we convert that glucose to pyruvate. Okay. One of the intermediates in going from glucose to pyruvate, this is respiration, is 1,3-bisphosphoglycerate. That 1,3-bisphosphoglycerate can come out of the cell, or come out of the pathway, excuse me, not out of the cell, but can get shunted out as an intermediate of the pathway and get converted to 2,3-bisphosphoglycerate. Okay, 2,3-bisphosphoglycerate is the same as diphosphoglycerate, so these things are the same thing, 2,3-BPG. Okay, it's abbreviation. So here we have a pathway of glucose metabolism, the breakdown of glucose into pyruvate, which is glycolysis. Okay, we'll learn this in the latter part of the course, all 10 steps. So just let me whet your appetite. One of those intermediates is 1,3-bisphosphoglycerate. It gets shunted out in the pathway, gets converted to 2,3-bisphosphoglycerate, and that 2,3-bisphosphoglycerate acts as an allosteric effector, shifting the oxygen binding kinetics of oxygen to hemoglobin such that oxygen will be dumped, that deoxyhemoglobin form is favored. Okay? So here we have 3 allosteric effectors, and one of them is actually intimately tied to the glycolytic pathway. So the breakdown of glucose in the red blood cell, this is more in the red blood cell, this phenomenon occurs. The breakdown of glucose makes an intermediary which produces an allosteric effector which influences oxygen binding to hemoglobin. So there is an intimate link between oxygen binding to hemoglobin and glycolysis. All right? So there is something in your book that talks about how people in high altitudes have different levels of 2,3-bisphosphoglycerate. It's related to oxygen altitudes and oxygen pressures. So people in the Himalayas, for example, may have more of this as opposed to people in Florida or in North Carolina. At high altitudes, your body adjusts by making more of this allosteric effector as opposed to less. So it has to do with your body physiology. All right? So the E and the T are not real molecules. Okay, E stands for exercise, and the T stands for temperature. Okay, these two things. If you exercise more, you facilitate an oxygen dump to your muscles. That makes intuitive sense. If you increase the temperature of the protein, that facilitates an oxygen dump of hemoglobin. Oxygen is going to get delivered to your tissues. All right. So this is a classic example of allostery. You notice molecules, CO2, H+, and this one, diphosphoglycerate, are coming in and influencing oxygen binding. They're influencing oxygen binding. This is called allostery. You could have positive allosteric effectors. You could have negative allosteric effectors. These are allosteric effectors that are actually favoring an oxygen dump. They're favoring hemoglobin to dump oxygen. They're favoring the T tight state of presumably all 4 subunits of hemoglobin. But what I hope I want to convey is that this tetramer, okay, of alpha 2 beta 1 alpha 2 beta 1 beta 2 alpha 1 this tetramer has cooperativity, and it can be regulated, so you can fine-tune this. You can fine-tune the oxygen binding. Okay, obviously you want, if you're exercising, okay, you want your hemoglobin molecules to deliver oxygen to the site. Okay, if you're sitting down and not doing anything, and and just kind of static, you know, the body can take some rest, and it could go through the normal basal level in terms of oxygen delivery. Okay, so we're talking about oxygen delivery not only to the muscle, but probably to the brain and to other peripheral tissues as well. All right. So once you get from a tetramer and hemoglobin, it becomes so complicated because we introduce other terms like cooperativity. We introduce allostery. Here are some 3 allosteric effectors. And the cell can now fine-tune the situation based on its needs. Hemoglobin is mostly associated with red blood cells. Okay, so I'm not going to go into specifics of this. CO2, if there's CO2 around, that produces carbamates. Okay, so you'll have to look at your notes to study that. And acid protonation actually is called the Bohr Effect. And there's some PowerPoint slides, and my notes talk about that into more detail. Right on cadet. This is a mnemonic. It's a memory device. Carbon dioxide, acid, diphosphoglycerate, exercise, and temperature. These 2 are not really allosteric effectors. They just cause the oxygen binding to shift. But these 3 molecules change and influence the oxygen binding. All right. So finally, we will talk some more about hemoglobinopathies. Okay, so a bunch of hemoglobinopathies. The most famous one is sickle cell anemia. And this one is really amazing because it tells you why amino acids are important, why you memorize those structures, why you need to know about tertiary structure, why you need to know about secondary structure. All right, so we're going to talk about that. It's a devastating illness. It causes the red blood cells to form a sickle shape. Okay, so our red blood cells are kind of this, I guess you'd call them a biconcave disc. But if you substitute, it's a single amino acid mutation, it's a glutamic acid to valine, in one of the subunits of hemoglobin. I believe it's the beta subunit, beta 1, but I'm not entirely sure which subunit. That one single substitution from the glutamic acid to a valine, okay, that actually comes from a mutation in the DNA sequence. It comes from a mutation in the codon. That results in a valine, and you would think that's an innocuous mutation, but you know now based on your side chains, this is a polar side chain, this is a hydrophobic side chain. What happens is that the hemoglobin molecules aggregate. Okay, aggregation diseases. And then because hemoglobin is so intimately associated with the red blood cell, all it does is has hemoglobin. It doesn't have anything else. All it does is glycolysis and hemoglobin. Okay, it actually kicks out its mitochondria during erythropoiesis, and it kicks out all its organelles, and its only job is to have an abundance of hemoglobin and perform glycolysis. But as a net result, this protein becomes a sickle shape. They clog all the arteries and veins, but the net effect is that the red blood cell becomes a sickle shape, a deformed shape, which as opposed to the healthy biconcave geometry. So a single amino acid mutation causes this. There's a bunch of other hemoglobinopathies. There's different forms of hemoglobin. Fetal hemoglobin is different than adult hemoglobin. And there's a bunch of diseases related to that. We'll talk about that during lecture. All right. So a very brief overview. Make sure you know about carbamates. I did not go over it into too detail here because this is supposed to be a very quick lecture. Make sure you know the influence of CO2, the Bohr effect, diphosphoglycerate, okay, these curves, and the fact of hemoglobin being a tetramer gives you cooperativity and allostery. So a lot more will be talked about during lecture, and a lot more information will be given in your notes. So please consult that. This is, again, a very quick overview that should supplement your notes. All right, thank you.